Okay, hello everyone. Welcome to the second live session for this course. So as I think a spreadsheet was already sent out and a few questions have come in, and then there were also some questions on the discussion forum which have been repeated quite a bit. So we'll try to address some of those. If you have more questions, please feel free to write here in the chat box now, and we can try to answer them. We have our TA here, Mr. Amaya, and so he will help us out in sort of reading out the questions, and I can try to answer them as they come in. So Amaya, you may want to start now.
Sure. One of the most common questions is when is the exam?
So I think the exam is on the December 19th. Is that correct?
Yes.
Yeah. Okay. So it's on December 19th. I think more details will come in as the date gets closer.
The next most popular question was people who are interested in getting a tentative feel of the questions of the exam.
Okay.
What?
Yeah. So the tentative feel is going to be very similar to what we have in the assignments. So there'll be both the MCQs with multiple correct answers, as well as the fill in the blanks type where a numerical or something is given and you have to solve that question and give the answer. So very similar to what you get in the assignments. So most of them will be directly from the slides. There will be some applied knowledge also that you may have to come up with your understanding. But nothing really will be very different from what you have already seen in the assignment.
We have a question on the chat box. Amulya is asking if we could give the students solutions for assignments in the problem. That's what I understand from the question.
Okay. Yeah. So I think this was also a question that I think somebody else was also asking in the discussion forum as well. So yes, if you need the solutions, please email the TA, and they will send it to you for sure. So yeah, if just drop an email to the TAs, or you can drop an email to me and I'll forward it to them, and they will send you the solutions.
Ashwini is asking if we need to refer any reference books in order to prepare for the exams.
You don't need to. Most of the exam, pretty much all of the exam, if you understand the slides well, you'd be able to solve. Of course, it's always encouraged to read as much as possible. So whether it's research papers or books or the slides, there were a couple of reference books that were given. But again, those are just optional. Those are recommended, not required. So you don't really need to go to the books. Everything that is present in the slides would be good enough to basically answer all the questions in the exam.
There was one more question in the discussion forum, and it read as, is hard corona formation synonymous to biofouling on implant surfaces?
Yeah, so biofouling is nothing but basically how different biomolecules or organisms, cells, come and attach to any new surface that they find. So as a result, any surface that you have will have protein that will adsorb to it, will have cells that will come in and bind to those proteins, may have bacteria that may come in depending on the where it is implanted. So the protein adsorption inherently is basically a subset of biofouling. So yes, so protein adsorption, whether hard corona or soft corona, all of those are part of biofouling.
Some students were interested in knowing the concept of core share nanoparticles. There was a question in one of our assignments and people were getting confused.
Okay, so I mean, I believe the assignment, let me see if I can just go here. So I believe that question in the assignment they're referring to was something that had a shell with some R1 here and some R2 here. And this was, of course, the shell, and this was the inner space where things could be encapsulated. So, for example, the drug could be here. So this is a core shell, basically meaning that there is a shell, could be made out of polymer, could be made out of metals, lipids, whatever it might be. Now, I think the question that was asked was to find a ratio between the volume of the core with respect to the volume of the shell. So the question was quite easy. All you had to do was find out the volumes. So we know that the volume of a sphere is 4 by 3 pi r cube. So if I say r1 cube, this will be basically nothing but the volume of the whole particle. Volume of the particle. No, r1 is the inner one. Okay. So let's say this is r2. And if I do 4 by 3 pi r1 cube, that is nothing but the volume of the inner core. So if this is already known, r1 and r2, then it's easy to find the volume of the shell. Volume of the shell is nothing but the volume of the particle minus the volume of the inner core. So shell volume is nothing but volume of particle minus volume of core, and so you can just subtract that, get the shell volume, and once you have the shell volume, you can find the ratios of whatever is asked in the course. Okay, let me go back to the video. Yeah.
One question read as, are ionizable drugs, by virtue of being charged, repelled from the cell membrane?
Yes, so okay, let's go back to this. Okay, so the question is, are ionizable drug because of their charge repelled by the membrane? So the answer is yes and no. So it depends on what you're referring to. So let's say we have a cell. We know that the cell has slight anionic charge. Now, if I have a charged drug that will come in, so let me draw a bilayer. So if I have a charge drug that come in and the charged drug is negatively charged, then it will of course experience electrostatic repulsion. But then if the drug that is coming in is positively charged, then it will actually experience an electrostatic attraction. Now, I believe what the person is referring to is the inability of the charged molecules to cross the lipid layer. So again, this lipid layer has a polar head group and a hydrophobic tail domain. So of course, any charged molecule can definitely interact with this polar head group based on whether it's positive or negative. But then because these are quite hydrophilic, they're quite polar, it will not tend to interact with this, so they can't really diffuse through this layer. And hence, the cell lipid bilayer is very good in terms of excluding anything that is charged. For them to go inside the cell, they need to go either through some receptors, through some ion channels, or something similar. So that's how they go in. So in terms of repelling, there could be an electrostatic repelling, and the other thing could be that even if it's interacting, it may not be able to cross the membrane simply because it doesn't have any hydrophobic domain that can pass through the hydrophobic layer of the lipid bilayer. Okay, let me now go back.
Going to the next question.
Yeah.
Pegylation has been shown to generate antibodies in vivo in humans. Why do we still continue to use PEG in several drug delivery approaches?
Okay, so that's an interesting question. The pegylation has been used quite a bit in the literature. The first generation of clinically approved products that came in, they were pegylated, and because they were pegylated, initially there was no concern that they may not have very good. There was no concern that they will generate any immune response because none of the animal data had showed this. But what happened is eventually when more and more experiments were done, and finally when a lot of this was starting to be used in humans, some of these were identified. So now, even though some of this has been identified, there are antibodies that are being generated. It's still only in a very small fraction of the population that actually shows this. So in a sense, still the large population is benefiting from these products. However, there is some population that does not benefit. In fact, gets more harmful for them. Now, the problem is there's no alternative to it right now, at least in clinics. There are several research that has been done by researchers all across the world, and they've come up with new polymers, new systems that could be used. But then, unless there is something that is clinically approved to replace whatever is out there, PEG is the only option. And given, as I said, it works in majority of the folks, it is still being used. So yeah, I think maybe a decade down the line, this may change when there are more products on the market. Then people would rather go for non-pegylated products, given that there is a chance that there may be antibodies produced. So I think in some sense this is still being continued because the research, especially with the human subjects, is a bit slow. And eventually, I think in a decade or so, if we find good alternatives to PEG, which are equally good or even better, and don't generate any immune response or antibodies, these will eventually get replaced.
Next question. Since both glidants and lubricants have a similar effect on the drug, what is the crucial difference between them?
There's actually really no major difference. Both of them are used to improve the flowability, as well as during manufacturing of these capsules and tablets. They are used so that machines can flow in powder, which can be then compacted into tablets or can be packed into capsules. So there's really no major difference. I would even go ahead and say glidants are part of lubricants. So yeah, that's basically that.
Sheetal is asking if she can fill the exam form now.
Yeah, I mean, so I think Sheetal is saying that she was unable to fill it by the deadline. Sheetal, I am not sure about this. I encourage you to write to the NPTEL admin folks and see if the deadline has been extended or if it's possible to register. Otherwise, this course will continue to be offered every year, at least for the next couple of years. That's the plan right now. So if that is not allowed, that then you can consider taking it the next time.
Next question. In a transdermal drug delivery system with a hollow needle, is it possible to clog the hole, which will result in a complete failure of the system?
Okay, that's an interesting question. So let's see. So I think what the question is saying. So you have these micro needles. And they're talking about hollow micro needles. So there's of course a hole, and inside these micro needles is all kind of drug that is being filled. Now it is possible to have, due to some or the other reason, have a possibility where some of these pores get completely clogged. But then, having said that, these micro needles are generally arrays of millions of needles, millions of very very small needles in micron size ranges. So even if one of these needles do get clogged, one of the needles, most of the other needles will still be available to continue to release the drug, and so it won't be a major factor. I mean, maybe 1 or 2% of the drug will be lost because of this phenomena. Most of the time, these are dissolvable microneedles that the field is moving towards. So even if it gets clogged, eventually all this polymer will start to degrade and release the drug. So it's not really a major concern that something like this could happen.
The next question is, what is the criteria for passing the course?
Okay, so the course is basically into basically there are two major parts to it. One is assignment and another is exam. Exam by itself carries about 75% of the weightage, so it is critical you do well there. But then assignments are also important, and assignments carry about 25% of the weightage. Since this is a 3 month or 12-week course, there will be 12 assignments. I believe there's about best of 8 or best 8 out of 12 is what will be considered to get that 25%. So it's important that both of them have a minimum threshold for you to pass. So yeah, that's basically the format of this.
Next question. Momita is asking if you could please explain the difference between intradermal and transdermal delivery.
Yeah, so intradermal is basically inside the dermis, inside the skin, and transdermal is through the dermis. So if you inject something where it's just localizing within the skin, if you give very superficial injection, that's intradermal, where it will lie in the skin. The skin has a lot of immune cells. There are dendritic cells, Langerhans cells, and all kinds of things that will come in and take up whatever is in the surrounding. And that is also a very good source to deliver things. And the other is transdermal, where you just penetrate that initial dermis layer and directly go below that, where things can continue to be absorbed in the system.
Next question. Amulya is asking if we could have a revision for numericals.
So Amuli, I think as far I know, it's difficult. Is there a particular numerical we could do it here? Otherwise, I encourage you to talk to TAs in the discussion forum, and they could, if you have any questions on a particular numerical, or even let's say 3 or 4 numericals, you could post it on the discussion forum, and I'm sure the TAs will respond with the solutions to those. Otherwise, if you can give us some heads up, maybe before the next live session, we could also just post some numericals in the Excel sheet that is circulated, and we could even do those numericals here.
Rupam is asking, what is the mechanism of release spray?
Okay, so I'm not really sure, Rupam. I'm guessing you're talking about these Volini type sprays, pain relief. Maybe that's what you are referring to. They are generally again relying on absorption through the skin, because most of the time they are sprayed over the area where there's pain, and most of the time they are some corticosteroids and other things that reduce the immunity as well as block the nerve sensation a little bit to prevent any major pain. So those things are absorbed through the skin and essentially blocks the pain receptors, so that the person who's suffering from the pain at the time could benefit.
So next question. Some of the students were interested in knowing what were the career options for students who would plan to take up drug delivery in the future.
Yeah, so I mean, that's a good question. There are a few options depending on what stage of the career you are. If you are an undergraduate currently pursuing the degree, you could think about maybe doing a master's or a PhD program in this to get more knowledge. There are companies that are now starting to come up, although not at a very high numbers in terms of requirements, but there are opportunities there. But most often you'll find there's a lot of opportunities in terms of research if you are really interested in something like this.
There, there was a brief confusion regarding the slides in the course. The TAs had uploaded a few slides, but then we had to go back the with another link. Just wanted to.
Okay. So I think a lot of you were asking if you could get the slides for whatever is being taught. So those slides were given now. So there was a Google link now that is active. So I believe that has already been posted in the discussion forum.
Yes.
So you could please. It looks like there was some initial problem in terms of access to that, but now everybody should have access to it. So just go to that link and you should be able to download whatever you need in terms of the slides.
That brings us to the end of
Okay.
questions in the discussion forum.
So yeah, right now that was all the major questions in the discussion forum. If you have any questions, please do post them right now. We'll wait another couple of minutes to see if there are any other questions. I hope you all are finding this course fascinating, and if you have any questions, please use the discussion forum. It's a very good forum for you to get any answers to whatever might not be clear from the lectures, and use this course not only just to get a credit, but also to sort of develop understanding. There are lots of things now which is hopefully will come up quite quickly in the actual field, as in there'll be a lot of clinical trials that are already going on, and then some of those will actually become a part of mainstream media or mainstream clinical trials. So I encourage you to spend some good time with this and see if you could also have some um, understanding which can last you for a longer duration than just to get a credit.
Rupam is asking if we could use drug delivery strategies for delivering drugs from herbal products.
So a lot of this is actually being used, Rupam. There are people extract reagents or biomolecules from plants, and then they use uh, um, those for various with the various drug delivery systems to deliver them for various applications. Of course, a lot of this herbal research has a bit of limitation that the lead molecule is not really known. So that creates a bit of a problem. Because if you have a single molecule, a purified molecule that you're looking at, it's easier for you to encapsulate, characterize, and go to the clinical trials. But then if you do not have that, then it's just a mixture of things. You don't know if there's an adverse reaction to it. You don't know if there is anybody who is maybe allergic to some of those things. So all those do compound this a little bit. But yeah, as long as the lead molecule is known, whether from plants or whether from animals, all of this could be potentially translatable.
Amulya is asking if you could have some theory revision classes.
Again, Amulya, I'm not sure if this is feasible with the current system, but again, we will have another live session. So if you could post as soon as possible when the live session is there on a specific topic, we could absolutely do this. You could also post it in the discussion forum, and as I said, TAs are very well equipped to answer any questions that you have specifically on any topic. So they will try their best to answer this. Okay, we'll wait maybe one more minute before we end this if there are no other questions. So if you have any questions, please do ask them right now. Okay, we will stop at this stage. Again, there will be another live session. Please do participate in it. If you send those questions early enough, then we can go ahead and answer all of them in a lot more detail. So I encourage you to please, whenever the next live session is announced, to please go ahead and post your question as soon as possible. And we can have a longer session also. There's no need for any inhibition in terms of asking questions. And yeah, good luck with your assignments, and I'll see you in maybe a month's time from now. Bye. Yes?